Welcome to Let's Get to the Marks, going through transport and exchange for A-level biology. This is a little question walkthrough, so pause the video now, have a go on a scrap bit of paper, then unpause and watch the walkthrough. For each question or each part of the question, just make sure you pause the video. Right, 6, figure 4, an image of a fish gill is taken using an electron microscope, a scanning electron microscope. Name F and name G, these kind of plates here that are down here, and name F, the structure that's holding those plates. Okay, let's go through the answers. So F is gill filament. A common mistake is gill arch. The gill arch is below, down here at the bottom of them, holding all the filaments off of. And G, these stacks are known as lamellae. You can call them gill lamellae. Now you need both answers for 1 mark for this one, so it's a bit of a brutal. Right, 6.2. Describe and explain the advantage of the countercurrent principle in gas exchange across a fish gill. So I've got a little video here you can click on if you want to know, but I'm going to go through the answer now. So the main thing about the countercurrent flow system is that water moves across the whole length of the gill in the opposite direction to the blood flow. So water's going in one way, blood's going the other way. This maximizes the efficiency of gas exchange. Now at A-level we need to explore how it ensures the steepest possible concentration gradient between the oxygen in the water and the blood, the oxygen in the blood. That way we always have oxygen diffusing from the water to the blood across the whole length of the fish gill. At no point is oxygen diffusing back from the blood into the fish gill, or is there a net diffusion of oxygen from the blood into the fish gill. So this means that oxygen is diffusing into the blood from the water across the entire length of the gill. The net movement is in that direction. And let's have a little look at some graphs that can help explain this concept. So some of you might be able to memorize the key points. For countercurrent flow, you want to say water and blood flow in opposite direction, 1 mark. This ensures the steepest or highest possible concentration gradient between oxygen in the water and the blood, and ensures that diffusion occurs across the entire length of the fish gill, always from water to the blood. They're your 3 points. However, I'm going to go through a little explanation of how we get to those points. This is the best little sketch that you should memorize, and it shows oxygen saturation on the y-axis on the left and distance across the gill on the x-axis of this little sketch graph. Now we can see that from the start of the gill, when the distance is 0, so right over on the left-hand side by the top of the y-axis, when the distance is 0, the oxygen saturation is at its highest in the water. The blue line here represents water. As the water moves across the gill, the blue line is getting less and less. Why is that? Because oxygen is diffusing from the water, from the blue line, into the blood, which is represented by this red line here on the graph. Now, obviously, the oxygen saturation, the blood is flowing in the opposite direction, so the blood starts from the other side of the x-axis. So 0 for the blood is on the other side of the x-axis. As the blood moves across the gill, the water is always at a higher concentration of oxygen than the oxygen concentration in the blood. So movement is always from the water to the blood. Now, you might say, what's special about that? Well, let's look if the blood and the water traveled in the same direction. They started at the same point on the gill and they moved together in what's called a parallel flow system. So we'd still have oxygen concentration on the left and oxygen concentration on the right. So if you think about it, the water is going to be giving up its oxygen and going down, and the blood is starting at the same point, so on the left as well. But at some point, the blood is going to gain more oxygen than the water, and the blood is going to start giving oxygen back to the water, a net movement of oxygen from the blood into the water, which means those lines will never cross. What will actually happen is, in a parallel system, they'll both level out here in the middle, where they reach 50% oxygen in the blood, 50% oxygen saturation of the water, and we get no net movement. They're in an equilibrium, but the result is that the blood does not have a high amount of oxygen saturation. So the parallel flow system is not a very efficient system for gas exchange in a fish gill. Sorry about all the knocking. That's my 3-year-old daughter banging on the door. Scientists captured a large number of 3 species of fish. The scientists measured the water depth where the fish were caught, the concentration of dissolved oxygen at all water depths, the mean gill surface area and mean body mass of each species of fish caught. The scientists calculate the ratio of their gill surface to the fish's body mass. The results are shown in Figure 5 and Table 4. Now, table 4 shows the relative oxygen concentration at different depths of water, or figure 5 did, sorry. Table 4 shows the 3 different fish species, and it shows the depth at which they were caught and their calculated ratio gill surface compared to the size of them, their body mass. Okay, so we can see here that oxygen concentration seems to go down with depth and then it pops back up again after like 750 meters. And then we can see for these 3 different fish gills, some of them have got a large surface area to body mass ratio. Let's check it out. 6.3, use the information in 5 and 4 to describe the relationship between dissolved oxygen concentration and increasing water depth, dissolved oxygen concentration and distribution of Plapoma fimbria. Oh my days. Right. Dissolved oxygen concentration and increasing water depth. So what we can see from that is that the oxygen concentration goes down in that first graph as we increase the water depth until about 750 meters, and after that it starts to increase again until about 1300 meters, where our graph sort of finishes. Okay, and that's going to get us 2 marks. So it's a description, so you're just saying it goes down, then it comes back up. And so these are description, because the command word in the stem is description. Look at the 3 fish species. What's the link between the 3 fish species and the depth of the water where they were caught? So we can see the first one, or the A fimbria that we're looking at, is caught in the range of 450 to 1280. It's not caught in shallow water. It's caught mainly in the deep water. So the dissolved oxygen concentration and the distribution of A fimbria. What we find is that A fimbria is found in a variety of different oxygen concentrations. I'd say from medium to low. So it's not found in any of the very high oxygen concentrations. So that would give you a mark if you said A fimbria is not found in depths of water with very high oxygen concentrations. We didn't see it at like 200 meters where the oxygen concentration's high. We found it mainly in a range of low oxygen concentrations, but it was found over a range of oxygen concentrations, as we can see. Okay, 6.4, using information from figure 5 and table 4, what can you conclude about the adaptation of the gas exchange surfaces of these species of fish? So what we can look at, the conclusion we can make here is the fish that operate at the low depths seem to have a large gill surface to body mass ratio. So obviously that would make sense, right? There's less oxygen in the water. They need to maximize the rate of gas exchange. So they're going to have a large gill surface to body mass ratio. So we want to say the fish in the lower oxygen concentrations, or at depths where the oxygen concentration is low, tend to have a larger surface area to body mass ratio. Surface area of the gill to the body mass ratio. Whereas one of the fish that doesn't operate at the very low depths, it has the lowest gill surface area to body mass ratio. That's the third fish. And that's going to pick your marks up. So you're just really explaining the link between the 2 graphs, which we can see that if you live in a low oxygen environment, you need a large gill surface area compared to your body mass. And there we go. That's the end of that question. Don't forget sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks.
You're still here? It's over.